Good afternoon. Today's topic is about the evolution of the digestion. It means that we will overview the evolution of the digestive system and also the evolution of the digestive processes. As a background material, I would propose for you the Veterinary Nutrition and Dietetics textbook. The next video will be a modified and enlarged version of a veterinary student's lecture. Our story starts at about 200 million years ago, when the carnivore dinos hunted and herbivores won. Here you can see on the left side a skeleton of a Tyrannosaurus rex with a body weight of 10 ton, strict carnivore, and right side you can see a Alamosaurus, a representative of the herbivores animals with a body weight of 40 ton. You can read a nice description about this story by Carl Sagan in his book The Dragons of Eden, or subtitled Speculations on the Evolution of Human Intelligence. He wrote about the extinction of dinos and the parallel development of the mammalians, and from the mammalians the apes, hominids, and later at the top of the development, the human mind. The Eumaya scansoria used to be a finding in the Gobi Desert, and it is accepted as similar to the presumed ancestor of all living mammals in the Cretaceous period. It was probably small, no more than 20-30 gram, nocturnal insectivore carnivore types, like today's mouse, or a screw or a mole, but living not underground. They could digest the exoskeleton of insects by means of chitinase enzyme activity. The residue of this enzyme activity can be found of the today's pig and apes. Their salivary glands produced mostly mucus saliva. The stomach used to be globular, and the small and large intestine short and simple. The cecum was present but small. It derived from the reptilian cecum. By means of secondary losses, no cecum can be found in bats, mink, hippopotamus, and giant panda. Yes, and here you can see some today's insectivores, the anteater, and also more and more supported by the WHO, the human insectivore. And a small picture give you how insect cleaner before and after ants, before and after a meal looks like. Today's insectivores representative, a mole. The shoes are similar. This is a mole digestive system. And what we can see? We can see a big stomach, which can be a reservoir for a little time. And as I've mentioned, a very simple digestive system and a virtual small cecum. From this type of digestion, the evolution started according to the field, according to the abundance of prey animals or plants in the environment. If no prey animals occurred sufficiently, but plant sources were present, there was a development in the direction of cecum and colon fermenters. So you can see A on the picture would be the common ancestor, and the timeline shows that it was 65 million years ago when the dinos finally extincted. And through the development in a territory rich in prey animals, more and more metabolic capacity were lost. And the real carnivore, cat-like Spheloidea, or the medium strict carnivores, the dog-like Canoidea, developed. Where sufficient plant was only present, the cecum fermenter glires, like the rabbits and hares, the cecum plus colon fermenters equidae, the horses, developed. By means of the fermentation in the large gut, they could maintain the life of a lot of microbes, and the microbes were able to ferment the plant fiber, and the developing volatile fatty acid by absorption through the gut wall gave a lot of energy. A medium type of digestion is the swine, where a lot of tuber and bulb was present, which are very high in, very rich in starch, with a high pancreas capacity, pancreas amylase capacity. And in some territories which were poor in nitrogen content, the plant was present, but owing to the arid climate, the protein content was low. The recycling of the nitrogen developed, and the ruminants and just ruminants, tulopods, developed. What happened a little bit more in details in case of the carnivores? You see that the carnivores' evolution started more than 40 million years ago. The first line are the pinnipeds, the water adapted carnivores, and the finnipeds are, and the fisipeds. Fisipeds are the field adapted carnivores. You see that we have two groups. The Phelloidea groups with the cat, hyena, and genets are strict carnivores. They lost a lot of metabolic capacity because by the body and liver and intestine of the prey animal they received all the vitamins and minerals in a ready form. The Canoidea group is, although they are taxonomically carnivores, but the digestive system and the digestive type from right to the left goes on from the carnivore to the omnivore. The weasel and the mink, similar to the genet, but the dog can more or less but digest some starch. The bear can be considered as a seasonal omnivore or fructivore or carnivore according to the season. And the raccoon is a flexitarian, is an absolute mixed based diet is used by the raccoon. The panda follow the separate line of development specialized to the bud eating. A little bit closer, we have to see the genetics of the domestication of the dog because it's also entailed a lot of changes. We can follow this process by mitochondrial DNA analysis. We can check back the time of the domestication, the loose domestication, which is approximately 100,000 years ago, and we can check the real ancestor of the domesticated dog, which is exclusively the wolf. In an Uppsala research, they compared the genome of the wolf and 69 today's dog breeds, and they found 34 genes as different, which changed in the dog after owing to the domestication. These genes are related partly with the carbohydrate digestion, higher amylase activity, etc., and partly with the development of the nervous system by enable dog with the timing and communication with the human. At the same time, structural variants in genes were associated with a human-like William-Burren syndrome. And this may underlie the stereotypical hypersociability in domestic dogs. On the bottom you can see the citation. So if you see the digestive system of a dog, you can make the conclusions that we have a big stomach, which is appropriate for more or less storage. A simple small and large intestine, and as I've mentioned, the cecum is very, very small, and to compare, you see the cat's cecum, this is even smaller. So they are rudimentary organs. Even in some cases, here you can see the digestive system of the mink or ferret, you cannot find cecum. And even I may add that even the peristalsis transit time is very, very quick in these animals. How can these carnivore or carnivore close animals use the short digestive system for efficient digestion and absorption? The way was the increase of the absorptive surfaces by villi and microvilli. In this slide, you can see the digestive system of a typical omnivore, namely that of a rat. Left side you see the result of a necroscopy, and right side you see the illustration made by an artist. You see that both the stomach and the cecum are large enough to allow a certain uh, reservation of the food in the stomach and also a fermentation of the fibrous material in the cecum. The small intestine and the large intestine are medium size compared to the carnivores or to the herbivores. And now we have to see a little bit closer the evolution of the monkeys as our closest relatives. And here you see the first step that most of the monkeys used to be herbivores, to be exact, because fruit trees were everywhere with major nutrients, but at the same time, fiber and secondary antinutritive plant substances were present. This is an advantage and disadvantage at the same time. Right on the slide, you can see plant cells depicting that the fibrous material, the plant fiber, chemically mostly cellulose, can be present and can be digested only by means of intestinal bacteria. The primitive monkeys used to be frutivore, and they have eaten and they eat even today leaves and buds too. So mostly frutifugous, and bud and leaf eaters are the apes too, but they are more selective and they can find very different type of foods. At least 10% of their food derived not from the plant origin, but by hunting and scavenging, but no more. And here you see the timeline of the main dietary shifts during hominin evolution. What are the hominins? The hominins are the lineage of up to and including extant humans following divergence from the human chimpanzee common ancestor. The time it was approximately 6 million years ago. Approximate dates of existence for human and chimpanzee bonobo lineage genera are depicted with using gray boxes. Timing of some of the important dietary transitions in hominin evolution is highlighted in orange and blue boxes. What were these changes? You can see these changes in the following. The main dietary shifts and changes were as follow. Approximately 4.4 million years ago, hominins moved from a forested habitat from the woodland to patchy forests to a savanna one. shift to harder and tougher foods allowed the development of large and thick emailed teeth to open large seeds. As an extra, the starch-rich underground storage organs of plants like the bulbs and corms offered new food sources. See the today's mole rat diet, for example. And 2 million years later, they started to eat meat by scavenging and hunting. And all the advantages of the animal food occurred, like the most valuable amino acid content, the energy concentration in the dry matter, liberated these individuals from the 20 hours searching for food. And they invented, for example, the fire. The time is not certain, 1.5 to million years or 800,000 years ago. The last is sure by finding. Fire and parallel with the fire, the cooking of the food sources increased the digestibility both meat and plants. At the same time, cooking produced byproducts, toxins, included the toxins with the potential carcinogenic potential. So, if you want to see more about this process, I would propose the last American scientist, an excellent article, The Hominin Next Door. Our ancestors shared the world with the puzzling Paranthropus. And these are the reconstructed form of these half ape, half human individuals. How we can imagine their food selection. As you can read in these articles of wood, you can see that the molar and premolar teeth are sheeted in an unusually thick coating of enamel. Chemical clues to the ancient diet gives also a solution, because the ingested food leave a chemical signature in the dental enamel. Namely, the plants can be classified to C3 and C4 plants according to their way of photosynthesis and the product of the photosynthesis. The C3 plants are from moderate temperature, and they are the cereals mostly, and the C4 plants are another cereals like the maize and the sorghum. And the maybe the biggest change and shift occurred 12,000 years ago, when the plant and the animal were domesticated. And instead of the previous hunter-gatherer communities, it allows a higher population density with a parallel reduction of food intake diversity. The agricultural products are high in starch, and the energy concentration is high, and this is the reason that it's influenced the insulin metabolism. The cereals and tubers has a low folate concentration too, which is disadvantageous to the health status. The enzymes undergone a genetic adaptation, which can be found by genome scan studies. The most important should be mentioned the lactase fluorescein hydrolase to split the milk sugar, and the salivary and pancreatic amylase activity, which increased, and as a plus, the N-acetyltransferase 2, which facilitate the detoxification of the toxins. At the same time, changes occurred in the sensory perceptions, taste and smell of the food. Based on all these, we can give a little critics to the Paleolithic diet, showing that we are not similar genetically 100% to the hunter-gatherer communities. Yes, and after that we have to see a little bit closer the evolution of the herbivores, which is also parallel with the extinction of dinos. You see once more that 65 million years ago the dinos died out. This is in the period of Cretaceous, and in the Paleocene and Eocene, etc., periods, the different groups, digestive groups of the herbivores developed. The horse-like, the pig-likes, the camelids, and the ruminants, parallel to the development of the cereals and the grasses. Yes. Here you can see a digestive system of a pig, which is a omnivorous type of digestion, with the exception that they adapted to the plant originated food of high starch concentration. Their amylase production in saliva and mostly in the pancreas is high, and they have a long small intestine with a great absorptive surface. But in territories where there was no tuber and another starch-rich product, animals started and developed a microbial fermentation in the cecum and in the huge colon. This is a cecum and colon fermenter animals like the equidae. Here you can see in situ picture of the huge large intestine of the horses. Yes. The question arises that is the horse equal to a big rabbit? Because it would be very easy to make a model animal for digestion trial. Instead of a big horse, you can work with a small rabbit. But unfortunately not, because the rabbit is only a cecum fermenter. And what about the donkey? He has long ears too, like the rabbit, like the Winnie the Pooh and the piglets, and the rabbit but unfortunately we cannot use. But what about the rabbit digestion? This is a very interesting way of the evolution because you can see here on this preparation that the stomach is huge, the small intestine is long, and we have a huge cecum with the appendix vermiformis and a colon proximale and a colon distale, but this is not large enough for the fermentation. Consequently, the rabbit developed a mechanism to make two different types of feces. One is going out like a normal feces, and another by a selection mechanism of the colon will produce a soft feces, which is practically a cecum content. If the cecum content as a soft feces is recycled by eating from the anus, the protein and other amino acids are saved. A very interesting side way of the evolution is the coipu or nutria, where no house tray on the colon proximale, therefore not perfect production of the two different type of the feces, soft and hard feces. So they are similar to the rabbit, but not the same. This is very visible if you open the colon proximale of the nutria. And here you can see the comparison of the coipu and rabbit gut. Left is the coipu in the center of the cecum, and the glet without house tray formed the colon proximale, and right you can see the rabbit's cecum and colon. In African territories in the savanna, where the plants had a high fiber and low nitrogen protein content, the rumination developed to economize the nitrogen metabolism, and the rumen and omasum, abomasum, and reticulum developed with a very long small intestine. The cecum and the colon is not so highly developed like to the horse. In the rumen, there is a fermentation of fibrous material and the production of volatile fatty acids, mostly acetate from the starch propionate, and from the fiber besides the acetate butyrate, as you can see the production rate on this slide. Volatile fatty acids, namely acetic acid, is produced in all the animal species. You can see here that the highest is in case of the horse. In the cattle, the highest is in the rumen, and in the pig, there is a little lactic acid production in the stomach, but this is not volatile fatty acid, and acetic acid production in the colon, but mostly in the cecum. Some animals lost enzyme activities, which substrate was ready to use in form in the food, and they had no reason to maintain this capacity. And in this way, the gulonolactone oxidase capacity lost in many animals, The delta 7 desaturase and the carotinase have last in the strict carnivores. But first, about the gulonolactone oxidase, because it's related to the human nutrition. If you classify the species according to the presence and localization of L-gulonolactone oxidase, we can see that monkeys, human, fruit, bat, guinea pig, and bulbul birds cannot produce, have no from this enzyme which would synthesize vitamin C from the glucose. The other mammalians, birds, reptiles, and amphibia have this enzyme in the liver and in the kidneys. And once more, without this enzyme, therefore, they need the vitamin C supplementation, the fish, and some insects. What are the deficiency syndromes in human, non-human primate, fruit eater bat, bulbul bird, guinea pig, fish, shrimp and krill? They are the small crabs. In animal with bones, ossification troubles, guinea pig, very visible symptom. Cage fatigue, even in birds, scurvy and bleeding gingivitis in human and guinea pig. And now some word about the strict carnivores like the cats. What would happen if a cat would receive only dog pet food? Dog is very close, many people think. It would this food low for the cat in protein, amino acids, and especially in arginine. The biotin concentration is not enough. The polyunsaturated fatty acid, namely the arachidonic acid, eicosapentaenic acid, and docosahexaenic acid, is not sufficient in the dog food for the cat, because cat cannot produce these polyunsaturated fatty acids because they lost the delta 7 desaturase. They have a higher manganese requirement, niacin. They also cannot produce niacin from tryptophan amino acid. And lastly, they have lost also the carotinase activity, so they cannot produce vitamin A from carotene. Therefore, they have to receive it ready in the pet food. All these would be scarcely would be deficient if you fed cat using a dog pet food. And the most important to mention that for the cat, the taurine is the essential nutrients, and lack of taurine will inhibit the bile circulation and finally will cause retinal degeneration, poor growth, poor reproductive performance. It means that low litter size, dilated cardiomyopathy, low birth of the newborn, birth weight will be low, and last but not least, compromised immune function. In case of human, the taurine would use part of the Red Bull because it has also antioxidant capability. Modification of all the previously told by the food and feed selections. Of course, we have mentioned the human But here you can see the grazing animals that we have concentrate selectors, immediate type, and roughage and fibrous eaters. All these will superpond to the previously mentioned. And lastly, you have to know a very interesting economy developed by the evolution. Hopefully, is not typical for human. This is the trophallaxis, which is the exchange of alimentary liquid, biological product, feces, secrete, saliva, either mutually or unilaterally. And also you can see another way that vomiting, regurgitation, or gastroesophageal reflux is present in the during the rumination too. But this is also a trophallaxis. The examples for that are the coprophagy, which is the intake of the own feces in case of energy or protein deficiency. Cecotrophy is the eating of the soft feces pellets derived from the cecum. Crop milk is a way how some birds are feeding their offspring, and the vomit or puke of the canidae. Please don't confuse it, the trophallaxis, which is a evolutionary development and normal phenomenon, with the allotriophagy and pica, which means that eating another which is not food, like soil or wool or the hair of other animals. To give a whole list of the other types of recycling and trophallaxis, you see that we have the saliva transfer, lip licking in case of rodents, shrews, genets. In this way, the young will receive enzymes and lysozyme. The mentioned crop milk in case of the pigeon, dove, and the parrot female. Pre-digested gastric contents by regurgitation, originally wolf dogs and jackals, and maternal feces in case of foal and elephant to receive the first microbes for the microbiome, and maternal feces and urine in case of dog, rat, and kangaroo. And finally, some illustration of the two types of feces in case of rabbit, guinea pig. The black are the high in protein, the content of the cecum, and the left side you see the normal feces. But this is also typical for the chinchilla, but here the cecum content can be found in the left. So the highest level is the cecotrophy of the rabbit, but you see that other animals developed similar mechanism. And finally, I will show you the miracle of evolution, a tiger rabbit, and by that I thank for your precious attention.